All right, yes. So let's go back. Deep learning in radiology, really popular. It's grown exponentially in the past few years. Okay. And this is an example CNN architecture. It's BGG19. It's quite old, actually. But this is just to show you essentially what it does. Right? So the reason why CNNs are so powerful is because they're able to approximate highly nonlinear relationships, approximate these relationships between inputs. The inputs could be images, could be tabular data, it could be like stuff like genomic data, and outputs, right? These outputs are typically classification or regression type outputs. But more recently, well, I shouldn't say more recently because the past few years had this. You can also predict images, stuff like that. So the way these essentially work is you have some input image, for example, and these input images are propagated through these series of mathematical operations. Now, those mathematical operations consist of things like convolutions. So the image is convolved with filters to output certain features of that image. You also have, let's say, pooling operations, which create some spatial invariance in the way it's processed, as well as more so to downsample the image so that you can handle that on your GPU memory, because we're limited on GPU memory, unless you have one of those really nice ones with like 48 gigs of memory. And then finally, you might have, or you will have linear activation or nonlinear activation functions. That's actually very important because that's why these work, right? We want to approximate this highly nonlinear relationship. Now, what we're focused on, what's most important in these networks are the feature maps and feature vectors, right? These features can be considered as just numeric values, just numbers that represent characteristics or attributes of this input image useful for the predictive task. So what I mean by that is you might have this little neuron right here have a large value, and that large value might indicate, hey, there's this weird bumpy texture in the liver, and that contributes to your decision of it being adequate or suboptimal in this case, right? Or you might have another neuron that's very small, saying that the morphology or shape of the liver is oblong or something like that, contributing to another, contributing also to the decision of adequacy or suboptimality. Right? So the main thing is we want to be able to interpret what these features mean, especially at this high level layer right here in the final fully connected layer. What do these features mean? Why are they important for determining whether or not this image is this class or this class? Now, specifically in the field of radiology, we actually face a major challenge, and it's that the interpretability or explainability of these algorithms, it's absolutely necessary if we want to translate these algorithms into clinical practice. It might be fine in like the tech industry, you just want it to work, right? But here we need to gain their trust. So in other words, these radiologists, a lot of the colleagues I work with, and clinicians, they want to be able to understand why CNNs are making the decisions they're making, and more importantly, why they're making incorrect decisions when they make incorrect decisions. Like, why are they classifying an image as not being a cancer when it's actually a cancer? So that can be quite dangerous. So in radiology, especially in radiological applications for deep learning, there are typically two popular approaches that are used to interpret classification and regression CNNs. So again, I'm focusing on these CNNs, not really the UNet type architectures, right? So the first is called GradCam. This is gradient weighted class activation mapping. The second is called saliency maps. So saliency maps have been around for a long time. GradCam came shortly after. There are some other methods that we can use, but these are by far the two most popular methods in radiology that I've seen in the literature. Now, there is actually a third that's quite popular. I've seen it in a few radiological studies. It's called activation maximization. The problem with that is it creates these images that try to visualize feature meaning, but those images look very surreal and dreamlike. So that can be difficult for a radiologist to interpret within that medium. But we'll talk about this more a little bit in a few slides. So GradCams. What are GradCams? Well, they're basically heat map visualizations. right? So there are heat maps that show the important regions of an image that are used to determine its decision, its given class output, right? It highlights these large areas of the image. So you can see that as we input this image into a network designed to determine if this liver is adequate or suboptimal for cancer detection, it says this large region over here is useful for determining that this liver image is adequate. Right? Now, we know that region contributes to the network's decision, but these grad cams are also, there are disadvantages to using this method, right? And mainly it's that sort of the subtle imaging characteristics, things like texture, or maybe the distribution of different pixel values across the entire liver, like diffuse imaging characteristics or diffuse disease characteristics. You can't really visualize it using this heat map right here. That information is lost, right? We just know that giant chunk is important for that decision. So that's useful in certain detection, object detection, classification tasks in the tech industry, where you see this giant blob over someone's face or over a puppy, because you know it's using that information. But what about the texture, right? Now, saliency map, on the other hand, saliency maps, they're somewhat more capable of identifying subtle characteristics. Think of this image as the derivative of the output with respect to your input image, right? So it shows the sensitivity of changes in your input image and the output. Now, these highlight pixels in the image that impact CNN decision, right, at the pixel level. But the thing is that the areas highlighted by the image might not follow a clinically discernible pattern, right? You have these diffuse areas that we're not quite sure what the meaning is. Is it looking at brightness, looking at texture, maybe some other pathophysiological feature that we don't even know? What's nice is these areas here highlight these darker vessels, and this area here, which is an artery, but what are all these activations doing?
Guys, can you switch off the lights in the car?
Yeah, yeah. Is that good? Now, lastly is activation maximization, right? This generates synthetic or fake images that visually display the features of the CNN. I mean, they look pretty cool, right? But they're so surreal and so dreamlike, it's very difficult to discern any sort of meaning in the context of radiology. So imagine creating a Picasso-like liver and showing it to a radiologist. They're not going to know what to do with it. This thing, I mean, you're introducing features into the image at this point. So it would be very confusing, very difficult for a radiologist to discern what the network is looking at. But we can improve this. Okay. So what we do to improve upon those limitations of those previously discussed methods is we propose something called feature interpretation using generative adversarial networks. It's FEGAN for short. So the whole job of FEGAN is just like activation maximization, except we generate realistic synthetic images that represent the meaning of the CNNs, what we call principal features, most important features. The whole point is we have this large feature vector, and we want to use its most important features by reducing its dimension. So this is just a quick summary of how this works, and then I'll go in depth into how it works. So first, we have our CNN. It's just a CNN that you want to analyze. You want to know how to interpret it, right? You take the CNN, and you take a set of images, propagate it through the CNN's layers, and then you extract that first, or that final fully connected layer I was talking about earlier, that just represents attributes of that image. You want to know what they mean in terms of appearance. Then we apply some dimension reduction technique. I say linear dimension reduction here because it's simple, straightforward. You can also use nonlinear dimension reduction if you want to, which essentially is what this network is doing to begin with. But we take these reduced features, then we input those features into a generative adversarial network, right? And using those features, we can generate a fake image that represents that one number. So if this number is high, this liver will look a certain appearance, right? And once that GAN is trained, then we can generate a synthetic image sequence across the entire range of feature values. So we can generate livers with small feature values and generate with large feature values. And as they transition, interpret that transition itself, show the movie to that radiologist so they can say, oh, that's what that feature means. So that's essentially what we're doing. So these are the methods. By the way, it's structured like this because I did this as a talk at SDSS, the Symposium for Data Science and Stats. So that's why it's a little formal. So let's say we have that network that we want to analyze, and we'll call it network C. That represents some separately trained, and I say specifically classification or regression CNN. And the job of this original network is just to map some input image, IC, to some output, YC. This can be a class output or just a continuous value, right? And we'll name just one of the feature layers, this final fully connected layer. And again, I choose this because it's the high-level features that are right before our decision. All right, so we want to understand what's going on. Now, FEGAN, the idea of FEGAN is actually quite simple. It's just a mapping. We want to map. We want to take these features and map the set of features back to our image space, right? And we do so using a GAN. That's what we do. So think of this is a function that sort of projects onto this manifold, and then now we want to map it back to that input space. All right, so this is FEGAN in a nutshell. We can probably ignore these details here. I might cover them a little bit, but so a GAN in general, this FEGAN in particular, is based on an image-to-image translation network. It's called Pix2Pix. It's actually quite old. It's been around for 4 years now, which is very old considering how fast the field is moving. But this was proposed by Isola and other authors. Really nice paper. Really revolutionized image synthesis. And basically, this GAN comprises 2 networks. There's 2 separate networks in this thing. So the first on the top is called a generator, and the network on the bottom, a discriminator. Right? So the generator's job is to take some input and predict or generate a synthetic image. That's all it does. Then that synthetic image comes down, propagates itself through the discriminator. The job of the discriminator is just to determine if that image is real or fake. That's all it does. So we predict a synthetic image, then we determine if that's real or fake. Now, this is actually an architecture called a UNet. It's actually very, very popular for segmentation and image prediction. Pretty much standard architecture for UNet, a standard classification CNN for discriminator. We did do some minor alterations, which I might go into later, such as turning off dropout and also replacing our transpose convolutions with nearest neighbors, if that means anything, right? But the key to take away here is the fact that we're going to generate synthetic images. Okay. So just a bit more notation, and this all goes specific to our application at this point. So if we let f represent some dimension reduction function, as I said before, we enforce linearity, so the transformation could be done by PCA, which I'll show you today. There's partially squares, independent components analysis. Honestly, any projection pursuit algorithm you could just use, and the results might change slightly, but honestly, we're still studying that. And then we'll let A represent just some form of additional information about the image. So this could be the actual just binary mask of the liver. It can be, say, clinical information, just anything that can help generate this synthetic image more accurately, right? It's just kind of boosting it a little bit. Then we take these features and auxiliary information, propagate it through that generator, which generates a synthetic image. And the way the discriminator works is it takes this synthetic image, ICS here, along with the inputs to the generator, and then ends up predicting whether or not that image is real or fake. Okay. So this is how we actually train the GAN, right? Remember, and I keep emphasizing this, all we want to do is generate a realistic synthetic image. So the way we train this GAN is adversarially. That's why it's called a generative adversarial network. Adversarially between the generator and discriminator. Okay. So we alternate basically between the gradient descent weight updates between these two networks. So for generator training, the first thing we do, we're only interested in making this predict real images. So we freeze this. This doesn't change. Our weights are just frozen, right? Then we send these features and auxiliary information through that generator network to create a synthetic image. Then send that synthetic image through this frozen discriminator network. And that discriminator determines if that image is real or fake. Now this is the key. If the discriminator predicts that this fake image is indeed fake, then through this adversarial loss, so we motivate it mathematically, through this adversarial loss, the generator changes its weights to make this image more real. So it can fool the discriminator into thinking that this fake image is actually real. Right? So we're motivating through this adversarial loss, this network to create a real image. Now, conversely, we still need to train this network, right? We're alternating. So during the discriminator training step, we freeze the generator, and we actually alter the weights or update the weights in two 1/2 steps. So the first 1/2 step is we send a synthetic image through, determine if it's real or fake. Except this changes because if it predicts that it is real, but it's indeed fake through its loss function, then it adjusts weights to be more accurate in determining whether or not it's real or fake. The second 1/2 step, it takes a real image, determines if that's real or fake, and if that image is predicted as fake, even though it's real, then it adjusts itself using gradient descent optimization, right? So we again alternate between these weight updates until this gets better and better at predicting or creating a synthetic image. This gets better and better at determining whether or not those images are real or fake. And as they keep getting better and better, these images become more and more real. Yeah.
You got a real image, goes, and then it decide that it's a fake. Who makes the decision to switch?
Yeah, no, that's a great question. So if I have a real image and it says it's fake, there's actually something called a binary cross entropy loss function. And based on that loss function, it's kind of like the mean squared error of regression, where you take the derivative of that loss with respect to the output, right? And then you end up adjusting the weights based on its gradient. So it's more like just a loss function equation that tells this network through gradient descent to adjust its weights properly. I feel like I botched that explanation. So, I mean, that was VGAN. That's pretty much it. That's the GAN. That's all it needs to do. It just takes features as input, generates a synthetic image, right? Now, of course, in theory, it sounded like a good idea, but we have to actually apply it to an application. So this is a bit more information on the liver application. So we wanted to assess its feasibility, right, using a previously developed CNN that was designed to evaluate contrast uptake adequacy of liver MR images. So just a bit of background, imagine that, you have an unhealthy liver, or you might have fibrosis. So they need to screen you every 6 months, just in case you develop a cancer called a hepatocellular carcinoma. So you go in, they scan you, and while you're laying in the scanner, they inject contrast. And in particular, it's called an intracellular contrast called EOVIST. I think Bayer makes it, maybe. But basically, it goes through your arteries, into your portal veins, and then your liver starts to light up because the cells in your liver start taking up the contrast, right? Now, the phase we're interested in is something called the HBP, or hepatobiliary phase, where all that contrast starts leaving your vessels, which makes your vessels dark and your liver bright. And because the vessels are dark, the cancers are dark. And it means we can see the cancer. Now, the problem is when a patient goes in and gets scanned, the tech sometimes acquires this image too early. And therefore, we don't know until the radiologist sees the image that, oh no, this was acquired too early, we need to ask them to come back in, right? So that's the whole point of this network. Or sometimes unhealthy livers, they don't have adequate uptake for cancer detection. So we want to determine if these images are suitable for someone to actually detect cancer. So the way it works is we acquire this volumetric MRI. We segment the liver using a QD liver segmentation CNN. That was developed by a colleague of mine a few years ago. Then we propagate it through this ResNet 50 CNN. A lot of you are probably familiar with ResNet 50 if you do deep learning. And there are slight alterations to this because we took 10 middle slices and propagated it through the network simultaneously in a batch for a batch update. But all you need to know is it's pretty much ResNet 50, and we have these features in the final fully connected layer that we're interested in. All right. So it's these features we want to interpret. So what is it about these features that determine whether or not this image is adequate? So what we do is we propagate 826 different liver volumes through the network. We take these features and then linearly reduce them using PCA to just the first 3 principal component scores, right? So in the end, we end up having 826 vectors of dimension 3. These are our features. We scale them to -1 to 1. That's just kind of standard for GANs. Specifically because at the output of the generator network, you have a TANH activation function, which is bounded above and below by 1 and -1.
And why 3?
Why 3? That was somewhat arbitrary. We actually looked into the percentage of variation explained by the different components. And honestly, it spanned such a large space that we were just experimenting on the most important. But I'll go into why. We want to increase the number of features, but as that space goes up, that cursor dimensionality, it becomes a harder space to explore. Yeah. So the input, finally, the input to this PGAN is a 224 by 224 by 4 channel array. Okay. So the first 3 channels are the features themselves. Literally, we took the number that was a feature and just repeated it 224 by 224 times. So it's an array that's a constant, right? So we have 3 of those stacked, and there's probably a more elegant way to do that. But the 4th channel is a liver mask. It's literally the tracing of a liver, but it's binary. So there's nothing about it. It's just white here and then zeros here, or 1s here and 0s here. Okay. As I said before, this is the decoder, right? It's the upsampling layers. Now we replaced transpose convolutions with nearest neighbors upsampling, and that was to combat this thing called checkerboard artifact. So the thing is, with transpose convolutions, you have this dilated kernel that you can involve the image with, and that can produce artifacts in these output images that just create these checkerboards, and I'll show you what I mean by that. But we just wanted to eliminate it, so we use nearest neighbors. Okay, training details. These are pretty standard. See, in the interest of time, I could skip those, right? It's just these are loss functions we trained for 500,000 steps. But these last 3 points are actually quite important for this to get working. So GANs are infamous for generator overfitting, right? They sometimes collapse and you're not able to predict images or generate images that are of good quality. So to prevent that, we augmented the data aggressively, and I mean aggressively, dynamically during training. So every time we sent an image for a weight update, we would randomly rotate it. We'd randomly flip it in all directions, so the liver would just appear everywhere. Shift it, zoom it. You can even add noise like to the case space, the Fourier space, or you can add noise to the actual image space. So that was just to prevent generator overfitting, and it turned out to be quite effective. We also infused the principal features themselves with random Gaussian noise, right? And this was to basically, instead of just regularize the image, we wanted to encourage smooth transition across our synthetic image sequence, right? We didn't want to show a movie and have the liver jumping all over the place. So that was to just enforce this. And I'll show you the results for that. And then finally, within the decoder of that generator network, there are actually dropout layers. Dropout layers just randomly drop neurons so that it prevents overfitting as well. But the thing about pix-to-pix is they actually keep those turned on during test time. And since there's random dropout, that introduces stochasticity. So every time you predict an image, the image is slightly different, right? We don't want that because we want this deterministic image generation. So we included dropout during training, but we turned them off during testing. Yeah.
How many will we need to have in training?
During training, it's a little complicated to say. So the input to the network was 826 liver volumes, but each 826, we took the middle 10 slices. Now we treated each of those slices as an independent, like training example. So you could think it's 8,260 multiplied by the however many augmentations. Yeah. So that was training. Then finally, during test time, we generate synthetic images, right? Have radiologists view these across the different values of the feature space. So we want to discern some meaning, elicit some meaning of this vector here from these images. And here are the results. They're actually quite interesting. I was very surprised when we first saw this. So small values of feature 1 were associated with this smooth liver texture. It's very smooth. There's no vessel visibility, right? You don't see vessel to liver contrast, indicating that it's probably not an adequate image. It's suboptimal for cancer detection. Now the thing is, a smooth liver texture can also be synonymous with poor liver function, meaning your cells are not healthy, so they're not absorbing the contrast well. And because of that, you have this uniform distribution sort of intensities. But as we increase that value, you see vessels start to appear. So what this indicates is that the network, this one principal feature, is focusing on the vessel to parenchyma or vessel to liver contrast to determine whether or not those images are adequate or suboptimal. For feature 2, this was actually more surprising. The first one, that's what doctors typically look at, right? But low values, you still have this uniform texture. But as we increase feature 2, the liver became even more and more unhealthy. It has this bumpy, and I think a radiologist said gross liver texture. Now this is indicative of something like scarring or fibrosis, like liver fibrosis up until cirrhosis. And this feature was actually significantly and negatively correlated with the outcome, right? The higher this feature, the more likely it was to be suboptimal. And this makes sense because fibrotic livers, they're unhealthy, so their liver cells are unhealthy. And finally, for the third feature, small values were associated with darker livers. And as we increased or ramped up feature 3, the liver became brighter and brighter, which makes also sense because if you have a healthy liver, it uptakes the contrast more and more and more, and your liver gets super bright relative to the dark vessels. So all in all, when we showed these 3 sequences to radiologists, they basically said, yeah, the first is tissue vessel contrast, so it's using that information. But more importantly, it's using nodular liver texture, which is indicative of fibrosis. That's something we weren't expecting, and tissue brightness. I say we were expecting this because radiologists, when they open up an image and see it, they look at whether or not the vessels are visible to determine adequacy, right? And I think implicitly they might determine the fibrosis with that decision. So what's nice is this gives radiologists some comfort because they're seeing, all right, these are the things I usually look at to make that decision, so I can rely on this network. Now, there are other things we looked at within the network. So interactions. The features actually interact, and this is kind of a bad thing because this complicates the interpretation, right? So we're looking into ways of just observing that main effect. But you can see here that as we increase feature 2, these large vessels appear. These almost look like hepatic veins or something like that. But then when we increase feature 1, the vessels disappear. right? So how do you interpret that? We don't know yet. Actually, these livers probably don't even exist because they have really nice vessels, but it's also super fibrotic. So just be aware of that. Looking into the need for auxiliary information. So we actually trained without that mask, right? That liver mask. And by training without the liver mask, we had to reduce augmentation. Because if we're flipping the liver everywhere, it's not going to know the orientation of the liver. What ended up happening is this weird morphology across the sequence. Since we reduced the augmentation, the generator started overfitting. So we have this diminished quality. And because of the transpose convolutions, right, we had this checkerboard artifact as well. Honestly, this is in hindsight, a bad experiment to show because it's showing all three things at once. We should honestly do one at a time. This shows the effect of adding Gaussian noise. Honestly, in my opinion, it's not that necessary, but you can see how when we generate the synthetic image sequence, it's like a vessel that jumps around, right? So by regularizing that feature space, we end up smoothing this transition. Now, I'm not sure how to measure this yet, but the smoothness here, it almost looks like the edges are dampened, which I don't like, because that makes it harder to discern a difference at either end, at the extrema of those movies. Another thing we looked into is the inclusion of that nearest neighbors upsampling layers as opposed to transpose convolutions. This is just the checkerboard artifact I was talking about. That checkerboard artifact, it wasn't prevalent, but it was present in certain images, and we just wanted to eliminate it altogether by replacing those upsampling layers with nearest neighbors. Right, and this is actually quite interesting. So we took the original CNN and we propagated those fake images to the original CNN just to see what would happen. So we increased the vasculature in this fake liver. And as we increased the vasculature, the confidence of it being an adequate image went up. Now, what's nice about this is it's in a way an intuitive validation that the synthetic images indeed represent the feature that the network is using, right, to make its decision. So it shows that this is not just garbage. All right, finally, comparison to other methods. So I showed you GradCam. It's a very large region. It's difficult to discern the specifics within that liver, like the texture. Saliency maps, we're not really seeing the meaning of the standard activation. Now, but brace yourself. This is activation maximization. So I actually ran it. And these are the two images that came up, right? Nothing like those surreal, deep dream Google images. Now, believe it or not, if I send this through that network, it has pretty much 100% confidence that this is an adequate image, right? That looks nothing like a liver, right? This has 100% confidence that it's suboptimal, right? In fact, they look like those, I don't know if people remember but those like audio stereograms of the 90s where you just kind of stand 4 feet away, go cross-eyed. But basically, these images, after looking at them for a long time, they kind of make sense, right? If you stand far back and see, you can see these long flowing lines, like long flowing lines here. And that can be construed as like a vasculature, like a vessel structure. Here you see all these short, round, chalky lines. That might be nodular texture, right? I'm just speculating, but it appears like it might be onto something, although I would never be able to understand or discern what was going on without seeing the FEGAN images. They can be useful, but a radiologist would see that and just see noise, right? In terms of how to use this method, I would never recommend using this method alone. I would say, look at the GradCam maps, look at the saliency maps, determine the region at which, or the region of attention. And if it's unclear what it's looking at, then look at these synthetic images to determine what they mean. So in this case, if this liver scored very high in the first feature, that first principal feature, then you know, okay, it's probably looking at the vasculature. to make its decision. Right? If it's scored very high, say on the second feature, then it's okay, it's probably a fibrotic liver. Or I shouldn't say fibrotic because definitively it might not be, but it has attributes of a fibrotic liver. So in summary, definitely has value in interpreting CNN features. We want to push this forward. It's been on hiatus lately, but we really want to publish this eventually. Some potential uses. It can be used to discover new imaging features, right? New features that we've never seen before, such as feature 2 for fibrosis. I mean, we weren't expecting that. Honestly, though, fibrosis has a clinical standard of care through elastography, which I think you should all look up the technology behind that. It's literally estimating the stiffness of tissue by vibrating your abdomen in an MRI machine. It's very impressive. You can interpret the relationship between other non-CNN features, right, and the outcome, which is useful. We did a study once looking at Euler characteristic curves. You can use those as input to the GAN and find out what the Euler characteristics tell you. In terms of limitations, as I said, the interactions are very difficult to visualize, and this goes up exponentially, right? Interpretation of it is actually independent of the original CNN, but I view this in a way as an advantage because it's an independent interpretation of the meaning of those features. So you could take that and pair it with your saliency map and determine what they mean. The difficulty in interpretation, or at least model fitting. Now, FEGAN randomizes weights as with any other network every time when you fit it. Randomized batches, but not only that, the sequences change at different steps. So we had to save the output every 50,000 steps to determine whether or not the GAN has collapsed. Collapsed meaning if the generator started overfitting and the images looked they just looked poor in quality. Future directions. So definitely explore larger feature dimension. We limited it at 3, but maybe we can go to 4 or 5. Not sure. Other dimension reduction techniques. So this is an example of implementing partial least squares to the features for linear dimension reduction to a network that used X-rays input to predict brain natriuretic peptide, which describes like heart health. It's a peptide that is correlated with heart health. Now, what we actually found is very interesting. These are actually fake. These are fake X-rays. These are synthesized by the network. And as we increase feature 1, you see this heart get larger and larger. And the subtraction image shows how this heart increases in size. Right? That's bad. This person has a large BNPP and is in danger of heart failure. Right? Not that it's a real person, but someone with a high value. Right? This one, actually, BNPP is biased for those with poor body habitus or those who are obese. So it actually captured the size of the person in the X-ray for the second feature, where this person's a bit wider than this person. This feature, we couldn't really discern the meaning. It focused more on the spine, it looks like, but we're not sure why. Could just be random. We'd have to rerun it a few times. All right, so just acknowledgements. This was work done in conjunction with, or in collaboration with Albert Chow over at UC San Diego, and some of the PIs over at the Liver Imaging Group at UC San Diego as well. References, if you're interested, they'll be recorded. Thank you.
Questions?
So I will ask a 2-part question. The first part probably don't have an answer because it's the same question every single time. Why did you choose those architectures?
Why did I?
And the second part is, how can you be confident? How sensitive are your results to the choice of those architectures?
Yeah, so choosing the architecture itself was just based on the architecture created in the original fix-to-fix paper. So I just took what they had and built upon that. I added some layers because, especially in the lung application, we had to change the resolution to be higher at 512 by 512. So we added more convolutional layers, but honestly, it was mainly the UNet's just so tried and true, we just rely on that. But that would be definitely a direction for future research, is looking at architectures and if it severely affects the output. Yeah. Did I answer both?
It's very interesting to see the meaning of those features. But I think it's still not enough to explain why don't classify make wrong classification of some image, right?
Mhm.
So I'm thinking whether we can develop a method like adding some feature, adding something to one viewer and change the like the classification result of the classifier.
I see.
Yeah, I also know there's one research field called solar pack. So they add some very small values to the whole image, and they can greatly change the classification result.
Yeah.
That definitely related to the like the unstability of the training of the classifier or the overfitting. I wonder if we can do anything to improve, or based on the understanding of the feature, how can we improve the current classifier based on the result?
I see. Yeah, no, that's a great point. So you're referring to, there's something called adversarial attack, where you could have an image that looks identical to the image next to it, but they add hidden information, literally add just data into the image, and it still looks the same, but it will completely change the prediction of the network. I mean, kind of one example of that is showing that noisy image. That was 100% confidence of adequacy, and it looked nothing like a liver. I don't know, it's difficult to overcome that unless you literally identify the specific features in the image. Kind of maybe using activation maximization, and then maybe subtract it out. And if the image looks the same, train with that, so that it diversifies its view. In terms of its use, I'd say if an image was incorrectly classified, but say the second score was very large and it was an adequate image, then we could say, oh, it thinks for some reason it's a fibrotic liver. Maybe we can go back and determine why. But yeah, that's the best we can do.
Any question from the Zoom?
Yes, can I ask a question, Kyle? Hi, Jose. How do you build your principal components? Like, how big is that matrix? You know, what data did you use to build the principal components?
Yeah, so the PCA was done on the output fully connected layer, which was of dimension 2048. So the actual PCA was applied to a matrix of size 826 by 2048. And then we only took the first three components to take the three scores associated with them.
You use only one image to build the principal component, or you actually use multiple images?
Oh, multiple images. So each image was represented in that matrix by that single vector of 2048 dimension.
Yeah. Okay. How many images did you use?
800.
Okay.
826.
Okay. And then what is the percentage of the variance explained? You say you use what, first 20 or some PCs?
I only use the first three scores. I think mainly for computational issues and just for exploratory purposes, but those first three components only explained 40 to 50%. And I think we needed over 100 to explain over 80. So I mean that space, it spanned a large dimension. And I'm not sure how to visualize that, because that brings up a great point, because sometimes even if it explains a decent amount of variability in the data, I don't think the signal to noise ratio is enough for the GAN to learn it, unless you have millions of images. So I think after the fourth or fifth component, it might not capture that variability. But we still have to look into that more.
In your GAN, did you only synthesize livers that have the condition?
So we synthesize, well, technically we synthesized all the livers within our data set. So we could synthesize the healthy ones as well.
You did that as well, so you
Yeah.
synthesized both.
Yeah.
Okay.
Just, yeah, any livers that span the entire domain space. And since the healthy and unhealthy were both included in the original training of the CNN, so they're just included in the GAN training as well. Mhm. You got a question?
I had a question about what you were describing for the dropout.
Yeah.
Like so when did you describe that? To remove that?
We kept it in during training. But what you can actually see is, so the authors of the original paper, by Asola, they included that because the original use of GANs was to generate synthetic data, maybe to supplement training, right? Generate synthetic data that would have a sufficient amount of variability. So like generating number digits, but very different looking number digits. So they included the dropout to attempt to create very different synthetic images, but it ended up just kind of changing them slightly. So it would be like a liver, and then the vessel might be morphed a little bit, but it would still look the same. And it was only apparent when we viewed them in a GIF that it was like warbly, almost like an elastic deformation. So when we removed that, it became smoother. So they even said, I think in the end of their paper, that was their attempt to create some stochasticity in their predictions, but it didn't quite work. There's probably some other way to do that.
Let me see if I understand this. We extract features from the image first.
Mhm.
And that's what the radiologists see. Right?
Mhm.
Generally,
Well,
right?
the radiologists see those images, but we extract the features.
The radiologists see that and make a diagnosis without looking at the feature extraction.
Mhm.
I'm asking, if you're a radiologist, then they show you the stuff, and then you say, okay, this, and he does a diagnosis based on those images.
Mhm.
Now you are extracting features from there that the radiologist actually could look at those features and say, hey, this helped me for my diagnosis. That's step one. But if I understand correctly, now that you are doing this post-processing with the adversarial network, then you will be able to show the movie to the radiologist, and the guy will say, yeah, this is this, and this, much faster.
Not necessarily. So this is more going on behind the scenes where the radiologist doesn't see, in this particular application, the radiologist doesn't see the image until after it's acquired. So a patient gets imaged and they may not see it till hours later when they come into work or log in. And this network, what we had planned was to determine the adequacy of these images at scan time. So a tech would say scan, it's like, oh, not adequate, suboptimal, and keep scanning until it was adequate. Now, the question is, how can we rely on this network? And the way we can rely on it is by increasing its transparency. So really understanding why it's making certain decisions. So this was more about just trying to understand the network itself. But in terms of helping a radiologist make a diagnosis, that is possible, but we probably want to develop a network that focused on like a diagnostic value now directly.
So my question is on a previous slide, you showed the pictures that look nothing like the liver, but one was considered optimal and the other one was suboptimal.
Mhm.
I wanted some, well, one was considered adequate. I wanted some insight as to why one specific one was considered adequate and why the other one was considered suboptimal. Like, is there some reason as to why that?
Yeah. So that goes to his point over here where during an adversarial attack, this shows that the network, we're interested in interpreting kind of looks at this set of features that's inherent in the image. not really looking at the features as we see it, but it actually identifies, possibly, I'm speculating, but identifies a pattern like this. And when it sees a pattern like this within that image, following all those convolutions, then it says, okay, it's adequate. That pattern is sufficiently present enough for me to predict it adequately. Now, again, I'm speculating, but when I see this, I think that abstract pattern it's looking for is like long flowing lines that appear as if they're vessel-like. Here, unhealthy livers tend to be like bumpy or have that texture, uniform sort of, yeah, and that's kind of what this looks like. It's really difficult to see, but can you guys see it? Like, there's like long flowing lines, and here it's almost like curly cues. But again, that's all speculation. I'm not quite sure, yeah. That's what was so upsetting. If you put an adversarial loss on these images, it might create a realistic looking suboptimal or adequate.
I was curious about using your methods for other applications. Would you be able to use a similar architecture? Is your architecture very specific to deliver and create some sort of transfer learning, or will we have to like retrain it from scratch?
Yeah, that's a great question. So architecture-wise, actually, we trained it from scratch, and you need a lot of data for GANs. They're difficult to train. But we used pretty much the same architecture as Pix2Pix. And Pix2Pix was trained to generate synthetic images of bridges and night and day and things like that. So it shows the capability of these architectures. They can actually apply themselves to many different applications, which is nice. In terms of transfer learning, especially in radiology, because images, your sample size can be somewhat scarce. Compared to millions of images, you had 800. Transfer learning's great. I would say always use transfer learning if you can. Whenever we try a new architecture or we fit another model, we try transfer learning and then we try it from scratch. Now, transfer learning, I believe, sometimes fails when you train on a set of images for a task that might be very simple. Now, if it's so simple, it might hit this local minimum within your optimization space where the features at the end of the network only look at one thing in your images. So when you retrain it on something else, you're already stuck in this minimum far away from what you would otherwise want to be in. But that's what's so powerful about the ImageNet data set. It's so diverse that pretty much no matter what, if you transfer learn using it, it's going to work. It's going to improve. Yeah.
So follow up, that's actually, I was going to follow up with that. If you train this on the ImageNet database, and then someone could download your pre-trained weight for your architecture for Thailand.
Yeah.
And then use it for their purposes. Does that exist already, or is that something that you're doing?
It's not, yeah, probably upon publication. There's actually another unit that we trained, and we just submitted the revision, but it's for lung registration. But we predict the deformation field of the lung using another unit. And we're going to post that online. Yeah, and that was pre-trained on 7,500 lung CTs. So it's very large data set.
That note, we're gonna thank our speaker one more time. To head towards Eureka for some reflections.